Alright, so in this video, we're going to be talking about pyrimidine metabolism. So under this, we are simply talking about how exactly are these pyrimidines formed. So in mammals and most lower vertebrates, they don't they do synthesize purines and pyrimidines. So those that synthesize purines and pyrimidines, we call them um, prototrophic. Okay. These are the mammals that are able to synthesize pyrimidines and purines. Okay. However, there are two types of pathways. Okay. The first one we call it de novo synthesis. The other one we call it the salvage pathway. Okay. So these are the pathways that are used to make pyrimidines. So under the de novo synthesis, the synthesis of these nucleotides begins with the metabolic precursors. So this is to say that this is starting with the raw fresh materials. Those are the ones that are used to make these. For instance, we have carbon dioxide, ammonia, ribose 5-phosphate, okay, and so on and so forth. But for the salvage pathway, it's simply a pathway that recycles those free bases and nucleotides that are released when nucleic acids are broken down. Okay, whenever there's a breakdown of nucleic acid, what the salvage pathway does is that it just uses the very same materials to make these pyrimidines. However, for the de novo synthesis, this one starts from scratch because it's using the raw materials. Alright, so looking at the structure of a pyrimidine, there are several things that we are going to look into. So this is the general structure of a pyrimidine, the generic structure of a pyrimidine base. So the nitrogen, let me just start from nitrogen number 1. So nitrogen number 1, that's right here, nitrogen number 1, as well as, so this is 1 2 3 1 2 3 4. So nitrogen number 1 and carbon number 4, as well as carbon number 5, 6, they all come from aspartate. And then when you look at this carbon that's right here, this one is donated by carbon dioxide, like you can see. Then carbon number 3, which is this one, is donated, it comes from the amide group of glutamine. Alright, so you're going to see all this as we go further in explaining exactly what happens during the de novo synthesis and the salvage pathway. So in the de novo synthesis, the pyrimidine nucleotide biosynthesis, the 6-membered pyrimidine ring is made first. This ring is supposed to be made first, right? And after it's made first, it's then attached to a ribose 5-phosphate. Alright, that's what happens in the de novo synthesis. However, when you look at the the next thing that we're going to talk about is exactly how that 6-membered ring comes about. So firstly, under the formation of the ring, the 6-membered ring, aspartate is our first raw material. So aspartate undergoes a chemical reaction where it's converted to N-carbonyl aspartate, and in this process there's an organic phosphate that's released. Okay, the enzyme that's used is called aspartate transcarbamylase. So after N-carbonyl aspartate is formed, this N-carbamylase is converted to L-dihydroorotase, which is this chemical, and the enzyme that is involved is Dihydroorotase. In the process, water is lost. Okay, this is a condensation reaction. So the formation of L-dihydroorotase the occurs in such a way that this enzyme, which is dihydroorotase, it removes the water. Okay, it removes the water from this N-carbonyl aspartate, thereby closing a ring, the ring structure. Okay, in the first place the ring was open, but now in this case the ring is closed, and it's called L-dihydroorotate. In the next reaction, there is what we call the formation of orotate from L-dihydroorotate. So L-dihydroorotate undergoes through oxidation. So this one undergoes oxidation. After it's oxidized, there's a transfer of hydrogen molecules, like you can see. And when there's an oxidation reduction reaction, what you notice is that the enzyme will always end with dehydrogenase. Okay, so the enzyme is dihydroorotate dehydrogenase. So all this is happening in order to form the chemical compound called orotate. Next up, orotate is converted to orotidylate. Okay, so the enzyme orotate phosphoribosyltransferase transfers a 5-ribosyl group, which is called the PRPP. It transfers this PRPP to the orotate, thereby forming the orotidylate, which is the new one. And all this is happening with the help of a magnesium molecule. Alright, so as simple as that. So now we have orotidylate. Now orotidylate is converted to uretidylate, which is UMP. Okay, so orotidylate undergoes decarboxylation. So decarboxylation is simply the removal of carbon dioxide from a molecule. So all the other types of pyrimidines are formed from this UMP. This is like our raw material to make the other pyrimidines, like UDP, UTP, CTP, as well as DUDP. They're all formed from UMP. Alright, so this is simply what happens. The next thing that we'll talk about is formation of UTP from UMP. So UMP gets phosphorylated, like you can see. There's a phosphate group is ripped off the ATP and added onto uridylate. So it gets phosphorylated in order to form UTP. Alright, so the enzyme that's involved is there's a kinase that's actually involved, and this kinase, let me just erase this. So there's a kinase. A kinase enzyme is the one that enables the phosphorylation to take place. Okay, so that uridine 5-triphosphate, which was formed in the previous reaction, it undergoes another reaction where glycine is involved, as well as ATP is involved, and the enzyme that is used is citidylate synthetase. So this is what aids the chemical reaction of converting uridine 5-triphosphate to citidine 5-triphosphate, which is CTP. This is the very reason why we said UMP is used to make all the other types of pyrimidines. Okay, so the next up we're going to talk about the formation of other types of pyrimidines. For example, DTMP. So like you can see, there is a movement, or rather there is a shifting of chemical reactions. We are moving from CDP going to DCDP, and so on and so forth. And as you can see, all these are the enzymes that are used to convert this. So this is dihydroxycytidine triphosphate being converted to dihydroxyuridine triphosphate. The enzyme is deaminase. Alright, so the final compound is DTMP, and the enzyme is thymidylate synthase, like you can see. So the DNA varies from RNA, in that RNA has uracil, whereas DNA has thymine. Alright, so the de novo pathway to thymine involves only ribonucleotides, okay, because we're talking about DNA. So this dihydroxythymine monophosphate, it can be it's synthesized by the methylation of DUMP, which is this. When DUMP is methylated through thymidylate synthetase, it's converted to DTMP. Alright, now this DTMP, the DTMP that's formed, it can be further it undergoes transphosphorylation. Okay, transphosphorylation. Okay, sorry for the spelling. So DTMP undergoes transphosphorylation. Alright, however, when you look at the de novo synthesis, okay, the de novo synthesis, like we said, it uses raw materials. So if this de novo synthesis is starting from scratch, this means that it's very expensive. Okay, a perfect example I can give you is if you want to buy a cake that costs 350 kwacha, right? It's cheaper than actually buying baking powder that costs about 180 kwacha. You're also buying milk that costs about 11 kwacha. You want to buy a tray, a baking pan that's probably costing maybe 100 kwacha. You're going to buy a tray of eggs costing maybe 60 kwacha in ShopRite. You're also going to buy baking powder costing 7 kwacha. You also buy salt, you buy sugar, and so on and so forth. So you find that you're going to spend a lot because you're using raw materials. That's why the de novo synthesis is quite expensive. So cells have therefore developed another pathway, or other pathways that can help to salvage purines and pyrimidine bases. To salvage is simply to just find a different, or rather an alternative way in which they can still make those purines and pyrimidines. So we have what we call pyrimidine phosphoribosyl transferase. Okay, this pyrimidine phosphoribosyl transferase, it catalyzes the formation of the pyrimidine nucleotide. It uses PRPP, like we had mentioned earlier. So this PRPP is the one that donates a ribosyl moiety. Okay, so this is about the salvage pathway. Okay, so now what you also need to note is that the pathways that degrade pyrimidines, okay, when these pyrimidines are finally broken down, if this is a pyrimidine, now it's broken down to smaller chunks and whatnot, they lead to the formation of ammonia. Okay, now this when ammonia is produced, it's not kept by the body in that state. It has to be converted to a different compound that's safer, for instance urea. So in this case, urea is then produced. because of the ammonia that has been produced by the breaking down of those pyrimidines. So pyrimidine nucleotides are then they get phosphorylated into free pyrimidines and a pentose. This is your pyrimidine, right? We have a phosphate group attached to. Okay, so this is your nucleotide. Okay, they get phosphorylated to form free pyrimidines and a pentose 1 phosphate. So they get um, separated into the individual components. Okay, so the next thing that we're just going to talk about is orotic aciduria. So these are is a type of disorder that's associated with pyrimidine synthesis. So the first one we call it type 1. This one is called type 1 orotic aciduria. Now, type 1 orotic aciduria is an autosomal recessive. Okay, so it means for someone to actually show traits of that disease, it has to appear twice in the genome in order for them to inherit it, or rather for it to show full expression. So this type that's autosomal recessive is a genetic disorder of a protein. Okay, it's a genetic disorder of a protein that we're going to talk about quite shortly. So the disorder is related to a protein that acts both as orotate phosphoribosyltransferase and as well as OMP decarboxylase. Okay, so this is what type 1 orotic aciduria is all about. It's an autosomal recessive disorder that affects the protein that acts both on orotate phosphoribosyl transferase as well as OMP decarboxylase. And we know that these enzymes have specific roles. So when they are affected, the orotate fails to be converted to uridylate. This orotate, it will fail to be converted to uridylate. Okay, there will be failure of it being converted to something else. So when this happens, the orotate tends to accumulate in the blood, and as it accumulates, it results to growth retardation in that child, as well as megaloblastic anemia. Alright, why megaloblastic anemia? It's because when orotate is not converted to uridylate, then the formation of these pyrimidines to form thymine, to form cytosine is cut short. Okay, hence the formation of the DNA is going to be affected, and the formation of red blood cells is going to be affected as well. They will not replicate. There will be no replication in the DNA. Alright, hence what we call megaloblastic anemia. Okay, so these red blood cells are big at the beginning and they keep on decreasing as they are developing, but when the process is cut short, they become big, and because they're big, they'll be too big to seep through certain tissues or cells, hence they'll start breaking. Okay, so we've talked about type 1. Now we're going to talk about type 2. Okay, so we're going to talk about type 2 orotic aciduria. Orotic aciduria. Okay, under type 2 orotic aciduria, it's also an autosomal recessive. Now this one affects the OMP decarboxylase. The decarboxylase enzyme, and it's also characterized by megaloblastic anemia. Like I had explained, and the urinary excretion of azidine is higher. The urinary excretion of azidine in higher concentrations than orotate. So azidine is going to be in higher concentration as compared to orotate. So this is what happens. Then the next type of disorder we'll talk about is orotic aciduria again. Okay, so we have orotic aciduria type 1, orotic aciduria type 2, and what we just call orotic aciduria. So orotic aciduria, however, is the one that accompanies the Reye syndrome, is the one that we're going to talk about. This one is associated with severely damaged mitochondria. This is the mitochondria, which is the energy powerhouse of the cell. It damages the mitochondria, and the mitochondria is then unable to utilize the carbonyl phosphate, which was one of the first reactants that we had talked about at the beginning of these chemical reactions. Let me just show you the first reaction we talked about was about making the carbonyl the N-carbonyl aspartate. Now the enzyme, the one of the things that I used to make it is carbonyl phosphate. Now carbonyl phosphate is going to be affected due to orotic aciduria. All right. So when carbonyl, the mitochondria is damaged, it's unable to utilize the carbonyl phosphate, which becomes available in cytosolic production of orotic acid. So carbonyl phosphate helps in the formation of orotic acid. So if the mitochondria fails to utilize it, it means it's going to be used to make more and more of orotic acid. Alright, so orotic acid is also associated with deficiency in urea cycle. Okay, like we said, at the end of this you must have ammonia. However, this is connected with deficiency in the urea cycle enzymes like enzyme transcarbamylase. Okay, so the deficiency results to excess carbonyl phosphate, okay, passing it to the cytosol where it's going to accumulate again. Now this carbonyl phosphate stimulates the pyrimidine biosynthesis. So the pyrimidine biosynthesis is the making of more and more pyrimidines. So in orotic aciduria, that's associated with the deficiency of the urea cycle enzymes, which is enzyme transcarbamylase deficiency, is the one which results in excess of carbonyl phosphate, and when there is excess, it passes to the cytosol where it stimulates more and more synthesis of pyrimidines. And you know that when pyrimidines are broken down, they give us ammonia. Alright, now we are also going to talk about drugs that are related to orotic aciduria. So one is allopurinol. Allopurinol, this one is an alternative substrate for orotate phosphoribosyltransferase transferase that we had talked about, and what it does is that it competes with orotic acid. So the resulting nucleotide product that's going to be produced also inhibits orodatylate decarboxylase, and this is what results in orotic aciduria. Okay, So I know I've mentioned quite a number of things, so let me just take you through once again. So one of the drugs that I used that are involved, we call them allopurinol. Now this allopurinol, it's an alternative substrate. A substrate is a compound that's used in this chemical reaction. So it's just an alternative, and this alternative is an alternative for the orotate. Let me write it down. Orotate phosphoribosyl transferase. Okay, it's an alternative for the substrate for orotate phosphoribosyl transferase. So this one, it competes with orotic acid. So the resulting nucleotide product also inhibits orotidylate decarboxylase. So whatever is happening in these processes is that they are affecting the substrates and the enzymes that are involved in the formation of these pyrimidines. Another one is 6 aziuridine. So 6 aziuridine, following its conversion to 6 aziuridylate, this one competitively inhibits orotidylate decarboxylase. Okay, so when orotidylate decarboxylase is inhibited, it leads to the increased secretion of orotic acid excretion, or rather orotic aciduria. So all these drugs are leading to orotic aciduria. So the next thing that we'll talk about are cancers. What type of cancers are related to pyrimidine biosynthesis? So cancerous cells have greater requirements for these nucleotides, okay, because this involves precursors of the DNA. For instance, if there is a mutation on the DNA base sequence where this segment has been deleted, a mutation may occur, okay, and if it's intense, it may even lead to cancer. So precursors of DNA and RNA are the ones that are affected, and consequently they are generally more sensitive than the normal cells to inhibitors of nucleotide biosynthesis. This DNA molecule is more sensitive to cancerous cells than any other cells. Alright, so the approach is that glutamine is a nitrogen donor, like we had seen in the beginning. It's a nitrogen donor in at least almost half a dozen of these separate reactions that we've been talking about in the nucleotide biosynthesis. So in order to stop this, it has to be inhibited. There is supposed to be an inhibition of glutamine amidinotransferases, which are aiding those chemical reactions. Okay, so the binding sites of glutamine and the mechanism by which the ammonia is extracted are similar to these enzymes. Okay, so most of these enzymes are strongly inhibited by glutamine analogs. These cancerous things that are cancerous cells. So what we're still talking about cancer. So the glutamine analogs are the ones that inhibit. For instance, we have what we call acivicin as well as azacerin. These are some of the glutamine analogs. So acivicin shows promise as a cancer chemotherapeutic agent. This can be used in treatment of cancer. Okay, that's why we are actually talking about it. Then number 2 is inhibition of thymidylate synthetase. Okay, so let me just take you back to a diagram where we were talking about thymidylate synthetase. Okay, should be way up there. Okay, so right here we had talked about thymidylate synthetase, and this is the enzyme that's aiding the conversion of DUMP to DTMP. Now, when this thymidylate synthetase is inhibited, as well as the dihydrofolate reductase, okay, these two, thymidylate synthase and dihydrofolate reductase, they provide the only cellular pathway for the thymine synthesis. These are the only ones that enable us to go from uracil to thymine. Okay, so why are we talking about this? So because they provide the only alternative pathway to produce thymine, they are quite very important. So fluorouracil inhibitor is the one that inhibits the thymidylate synthase, whereas methotrexate, this one is, and as well as aminopeptide, this one is the inhibitor for dihydrofolate reductase. Okay, so these are some of the inhibitors. Now you know that when these when thymidylate synthase and dihydrofolate reductase are inhibited, it means the pyrimidines will not be made as they are supposed to. Alright, so in summary, what you're going to learn, or what you need to expect from this lecture, is that you need to know that the pyrimidine is made first by making the ring first, and then it's attached to a ribosyl 5-phosphate. Okay, that's point number 1. Then the amino acid glutamine, as well as carbon dioxide, aspartate, these are the ones that furnish all the nitrogen atoms, as well as carbon dioxide. This one is giving nitrogen, this one is giving carbon dioxide, as well as nitrogen. Alright, so the first pyrimidine to be formed is UMP, like we said, and from this UMP, this is where all other nucleotides are synthesized. So that's it for this lecture.